We're very pleased to present immune responses to gut bacteria predict disease onset in a type 1 diabetes prevention trial by Quin Xie. She's a grad student, a PhD candidate in the Department of Medical Biophysics, where she combines single-cell analysis, machine learning, and statistical modeling to study the role of gut microbiota in the progression to type 1 and type 2 diabetes. Her PhD project revealed, sorry, still letting people in, her PhD project revealed the association between immune reactivities to gut microbes and the treatment efficacy to the first FDA-approved type 1 diabetes prevention drug. More recently, she's been focusing on understanding the immunometabolic changes in obese adolescents that lead to type 2 diabetes risk. She's grateful to have been supported by scholarships and awards from multiple sources, including the Federation of Clinical Immunology Society's Center of Excellence, Banning and Best Diabetes Center, and Hilda and William Courtney Clayton Pediatric Research Fund. And her long-term goal is to understand the mechanism of host microbiota interaction to provide insight on developing biotechnology tools and novel therapeutics. And just to sort of reiterate, she's a PhD in the candidate in the Department of Medical Biophysics at University of Toronto, in the lab of Dr. Jane Danska. So thank you so very much for joining us. We're really excited to hear your presentation, and if people are interested in asking questions, please feel free to drop in questions into the chat. We will field them as we go along, and we'll take questions at the end.
Thank you so much for the kind introduction, and it's a great pleasure for me to be able to present my project here to an audience that's interested in this disease. And I know that many of the people here might know a great deal of this disease already, but I would just like to start with a bit of introduction on type 1 diabetes so we are on the same page. It is a T-cell mediated autoimmune disease currently affecting over 9 million people worldwide, and it has been shown to have a great genetic component. Previous genome-wide association studies have identified over 80 risk loci that are associated with the disease. And here I am showing a Manhattan plot, which is a common output of these kind of studies, which shows that many of these genes are actually immune-related. And among these genes, HLA-DR is the one with the largest effect size, which conforms to our understanding of type 1 diabetes pathogenesis, because variation in genes encoding these HLA molecules will affect antigen presentation to T cells and therefore change the peptide binding propensity of these molecules and therefore bias the T cell repertoire. So it is very unfortunate that beta cell death and the loss of insulin, which are characteristics of type 1 diabetes, is irreversible. So once patients are diagnosed with type 1 diabetes, they are likely dependent on lifelong treatment with exogenous insulin and are potentially at risk for many complications if they don't take care of their lifestyle. But thankfully, because of research effort on type 1 diabetes genetics, as well as the search for prognostic biomarkers, we now have the opportunity to find individuals relatively early in the disease process so they can benefit from the treatments that preserve residual beta cell mass as well as delay disease onset. So as we can see here on this diagram that's put out by TrialNet, which is a consortia that studies individuals at risk for as well as who has been diagnosed with type 1 diabetes, the onset of clinical symptoms is actually considered stage 3 of the disease. And what this means is that if we intervene early enough, beta cell functions in at-risk individuals can actually be preserved by disrupting the autoimmune process. And so this is exactly what TN10 prevention trial was for. In this trial, individuals who have not been diagnosed with type 1 diabetes were given a T-cell targeting immunotherapy. And they were recruited into this trial because they show multiple signs of risk. So for example, they have first-degree relatives with type 1 diabetes, and this, in particular, most of them carry the high-risk HLA-DR haplotype that themselves alone carry over 50% of the genetic risk. As well, they have multiple types of islet autoantibodies circulating in their blood, and that indicates ongoing autoimmune process. And so, as I mentioned before, these individuals were given a single dose of T-cell targeting treatment, and this is called teplizumab. It is a monoclonal antibody that disrupts T-cell function by binding to part of the T-cell antigen receptor called CD3, but does not kill the T-cells. And the primary outcome of the trial is the time elapsed between the randomization into either of the treatment arm and type 1 diabetes diagnosis. And from here on, I will refer to it by time to type 1 diabetes. And it is actually the first time that we are able to successfully delay the disease of the onset of a disease using immunotherapy. And so what I mean by that is very clear if we look at the Kaplan-Meier curve on the right. So it shows that at any given time during the follow-up of the trial on x-axis, the proportion of individuals that are free of type 1 diabetes is higher in the teplizumab marked in blue than the placebo group marked in red. This is such an exciting result that FDA approved this drug about a year ago now, exactly for the purpose of type 1 diabetes prevention. And so this all are very exciting, but as is commonly seen in many other immunotherapy trials, there's response heterogeneity. And what I mean by that is that despite the fact that individuals receive the treatment at the same time, there are some that will have earlier onset of the disease than others. And so in the context of this trial, we found that individuals carrying HLA-DR4 haplotype will have greater delay in type 1 diabetes onset than those without. And so a gap in knowledge is to identify individuals that are likely to respond to the treatment. And we hope to take an approach that will either shed light on the mechanism by which teplizumab takes effect, or find additional markers to help us guide the use of this very promising immunotherapy. And here, our reasoning is that factors that are associated with progression rate to type 1 diabetes clinical onset, will also influence whether an individual will respond to the treatment or not. And so we proceeded to identify these factors. And as I mentioned before, HLA-DR is a major genetic risk factor. So definitely that plays into how rapidly the disease progression will go. But what's interesting about this disease is actually its changing epidemiology, which points to potential environmental factors in influencing disease progression. And specifically, if we look at this plot here, it shows that over the past 60 years, type 1 diabetes incidence has been increasing worldwide. And we definitely see geographical differences in incidence rate being more common in higher income, more developed countries, as well as having earlier age at disease onset, peaking at 5 to 7 years of age, as well as pre-puberty. And changes in disease epidemiology in such a short time frame really points to the existence of environmental factors that are either acting independently or in interaction with genetic factors that bring about changes in disease incidence. And what are some of these examples? We might think about improved public health, diet, lifestyle, as well as antibiotics use. And interestingly, all of these factors will influence the gut microbiota, which is the community of non-pathogenic microbes that are residing in the gut of the host. And we know for sure that gut microbiota has important roles in shaping immune system development and function. And so this might be a linchpin between the genetic determinants and the environmental modifiers of type 1 diabetes risk. And so an intuitive way of studying the gut microbiota is to sequence them. And indeed, we have numerous well-done deep sequencing studies on thousands of babies at risk for type 1 diabetes, but we do have difficulty pinpointing specific microbes or taxonomic composition that are associated with progression to type 1 diabetes. In fact, I think we haven't reached consensus in the field on what a normal gut microbiota should look like. And so we took a different approach. We reasoned that we know that gut microbe-derived antigens and products will be able to reach systemic lymphoid organs, either via general circulation or via antigen-presenting cells, and therefore induce the production of non-pathogenic serum antibodies. And our hypothesis is that if gut microbes are able to influence autoimmunity against the pancreas, which is a different tissue from the mucosa and the associated immune system, tissue, then we would be able to detect patterns of these immune responses in the systemic circulation. And so by looking at the immune detection of microbial antigens, we hope to be somewhat more informed about type 1 diabetes risk. And so we invented a method to examine host immune responses to gut microbes. To do that, we add bacterial targets to serially diluted serum samples to allow serum antibodies to bind to the surface of the bacteria. And then we use flow cytometry to quantify the percentage of bacteria bound by serum antibodies of different isotypes. And then we plot the percentage of antibody-bound bacteria against log dilution flow to calculate a response index for every combination of bacterium and isotype in each sample. And we applied this method to study a cohort of pre-type 1 diabetes individuals. And we found that these patterns can indeed distinguish between individuals who eventually progressed to type 1 diabetes from those who did not. And these associations are contingent on HLA-DR haplotype. And now the question is, can we find anti-commensal antibody responses that are associated with outcomes of interest in the TN10 cohort? And by outcomes of interest, I mean either responses to teplizumab or time to type 1 diabetes diagnosis.
Quick question, Quinn, from Boston. What age sample were collected and which stage of disease development?
Yeah, so that's a good question. I was just going to answer that in the next slide. So the median age of these individuals are 14 years old. 14 years old in both treatment arms, and they are considered stage 2 of the disease because they have multiple types of islet tissue antibodies, but they do not have diabetes. I hope that answers the question. And so we use this method to analyze 228 serum samples that are collected before and after randomization at multiple time points. And we looked at responses to 31 strains of gut microbes from 15 species, and they cover for most abundant bacteria phyla in the human gut microbiota. So this project, this paper has been published on Science Translational Medicine quite recently. So if you're interested in this talk and you want to know more about it, you can find it online. But for the interest of time here, I have shown some. I'll show some selected results here. So this is a histogram showing the distribution of IgG2 response to a bacteria called Bifidobacterium longum. It is a quite common probacteria. And this is the distribution from all participants across multiple time points. And we see that there's a bimodal trend in the distribution. So we dichotomized that for the purpose of downstream analysis using the lower point between the two modes. And here are the same Kaplan-Meier analysis that I have shown before. And these samples are from the baseline only and stratified based on whether they have low or high IgG2 response to B. longum. And they are color-coded such that individuals from teplizumab group is in blue, and the placebo group is in yellow, and the shaded area is the 95% confidence interval of the survival curve. If we look on the left here, for individuals with low baseline IgG2 response to B-longum, there seems to be much overlap between the shaded area, indicating that these individuals do not significantly benefit from the treatment. Whereas if we look on the right, for individuals with high IgG2 response to B-longum at baseline, there's a much clearer separation between the two treatment arms, indicating that there is a significant difference in terms of response to teplizumab. And we also used Cox proportional hazards model to estimate the effect of treatment arms and IgG2 response to B-longum. And we showed that there is significant association with time to type 1 diabetes, as is shown in the bottom right, with p-value less than 0.05, 0.05. Yeah. And we initially restricted our analysis to baseline samples because we were interested in predicting treatment response. But actually, one of the features that we found about these anti-commensal antibody responses is that they are very stable over time. So we repeated the analysis in samples collected 6 months after randomization. And what we can see is that the trend is largely the same. So if we look at individuals with low IgG2 response to B-longum on the left, again, not much separation between the two treatment arms, whereas on the right, there is a much clearer separation, and the treatment effect as well as this anti-commensal antibody response is again significantly associated with time to type 1 diabetes diagnosis. And so the previous example is an anti-commensal antibody response that's associated with outcomes of interest in the entire cohort. And the next example that I have chosen here show that commensal antibody response patterns can display interaction with HLA-DR haplotype in their association with time to type 1 diabetes. And here I am showing the distribution of IgG2 response to D in vistas from samples that are collected 6 months post-randomization. And again, they're dichotomized for downstream analysis. Here, this plot might seem a little busy here, but it's the same type of Kaplan-Meier analysis, but now they're stratified both based on HLA-DR for haplotype and IgG2 response to D in visas. So on the left here, we have individuals carrying HLA-DR4 haplotype. On the right, individuals without. The dashed line are representing the individuals with high IgG2 response to D in visas, and the solid line for those with low responses. The color coding is the same as before, and the confidence interval has been omitted for the clarity of the display. And so what we can see.
Oh, sorry. From Colleen Cutcliffe at Pendulum Therapeutics. She's just asking, I can't tell if it's just the visual on this slide and previous one, but it seems like it's the placebo that looks different, not the treatment arm.
Sorry, just to clarify,
thank you.
It's the B-longum data. So I don't know, do you mind flipping back to that? And again, I'm looking at a small screen, and so it might just be the visual. If you just, this slide and the previous one, where you're trying to show an association between the levels of B-longum and then kind of I think what you're showing is that there's a bigger difference between the placebo and the treatment arm when people have high B-longum.
Right.
And so on this slide and the previous one, definitely it looks like there's a bigger difference between placebo and treatment arm on the right-hand side than the left-hand side. But it looks like what's changing, though, is actually the placebo arm. But I can't tell it's visual because your x-axis, I think maybe the days are not. Anyway, can you just talk to that for a second?
Yeah, I mean, these are statistical analysis, so I try to not read too much into that. But if I were to give an explanation, I would say that, taking this drug kind of put a pause on the autoimmune process in these at-risk individuals, whereas having being in the placebo group means that they're just progressing to type 1 diabetes. So that might be the reason why you're seeing more changes in the placebo arm in terms of survival or time to type 1 diabetes. But in the original paper, we've also shown that, these anti-commensal antibody responses are not associated with the magnitude is not associated with the teplizumab treatment itself.
I see. Okay, I'm sorry. I just didn't understand. Right. So what you're saying is it's actually not got to do with the intervention, which is what I was sort of looked to me like. But what you're saying is that the people with the high levels of B longum that aren't given anything are progressing more rapidly.
Yeah, more. Yeah.
And so when we think about, and I understand we don't want to draw too many conclusions from a correlative study, but essentially what this would point to is that you want low B longum levels. Is that right?
Well, we will want high high IgG2 response to B longum for individuals that are going to respond better to the therapy, because what we see is that like as a predictive marker, I guess, for individuals with lower IgG2 response to B longum, there isn't much difference between the two treatment arms. Whereas if you give this treatment to individuals with high IgG2 response to B longum, they're more likely to respond to the treatment. Does that make sense?
No, but it's okay. We can keep moving forward.
Okay.
Thanks for the point of clarification. Yeah, I think the whole focus is really to identify those who would best benefit from teplizumab in this context.
Yeah.
Right.
We're really focusing on like prediction in terms of both type 1 diabetes risk as well as treatment response to teplizumab. So these anti-commensal antibody responses, they probably, like, in terms of their immune exposure to these gut microbes, it's probably much earlier in life than when they start to develop autoimmunity to like the islet tissue. So we're really trying to like predict the response as well as type 1 diabetes here using these anti-commensal antibody responses.
Yeah, and I would just comment that a beautiful paper came out from Diabeologia, June 2023, infant gut microbiome composition correlated with T1D acquisition in the general population, the ABIS study. That was Johnny Ludvigsen and Eric Triplett, and they really showed that, these gut microbial biomarkers in 1-year-old infants associated with progression to future type 1 for up to 20 years before diagnosis. So things are going on, as you just mentioned, quite a bit, earlier than the presentation or the march closer to diagnosis.
Well, I guess maybe if you just look at this, and you can tell me if I'm looking at this data the wrong way, and this is not my specialty. I'm not in type 1 diabetes, so I'll give all those caveats of coming fresh to this. But if we look at, day 1095, on the left-hand side there, it looks like the blue line is sitting somewhere maybe around 0.6 to 0.7, and the yellow line is around 0.55. And then if we look on the right-hand side here, that same day 1095, the blue line looks like it's, maybe around 0.55, but it's the yellow line that looks like it dropped significantly to, whatever, 0.3. So that's really my question, is that it looks to me, and again, I would want to spend more time with this data. I could go look at the paper, but it looks to me like the intervention arm, what I'd love to see is an overlay actually of the intervention arms and the placebo arms on one plot together, where you're comparing high and low. So yeah.
Can I respond to that?
Yeah. Thank you,
Eric. It's just that, Eric. Remember, on both slides, there are different people on both slides with likely different genetic risks. So I don't think you compare placebo on one side to placebo on the other. They're really separate experiments. But it is clear that the difference is much greater or is significant when the bifidobacterium is higher. So
I definitely agree with that, Eric, but I guess my question is the difference is greater, that's true. And maybe you're saying, look, these people are so different that you can't compare placebo to placebo.
You can't really compare placebo across the two groups, in my view.
Okay.
Sorry, I wanted to just clarify that these are same individuals, but they're from two time points. So like, I guess you could compare that. But in the original paper, we show an overlay of high and low IgG2 response to B longum on the same graph at baseline. So you can see a much clearer separation between the traces if you're interested in the original paper.
Yeah, I mean, I can go look at it. I'm happy to go look at it. It's more that I'm trying to think about, you know, from a translational standpoint, you know, are you really trying to look at improving the efficacy of the treatment, or are you looking at, you know, an ability to improve efficacy or whatever, help people that don't go on treatment? So that's what I'm trying to get at by looking at the data. But I'll go look at the paper because it's maybe going too fast here.
Right. Yeah. Okay. Thank you.
Thanks.
Great discussion.
And so just continue with the explanation here. If we look on the left, we see a much clearer separation between the traces. And that indicates that for individuals with HLA-DR4 haplotype, if they have higher IgG2 response to D. inviscis, there will be a higher risk of developing type 1 diabetes more quickly. And in this subgroup, teplizumab is effective at delaying type 1 diabetes diagnosis, whereas on the right, the separation is less obvious and there's no clear association of IgG2 response to D. inviscis to treatment effect or type 1 diabetes. And so, as I mentioned before, HLA-DR4 is associated with treatment response in this trial. So together with this information, we think that anti-commensal antibody responses have additional value in identifying individuals that are likely to respond to the treatment. And if we want to quantitatively evaluate the added value of anti-commensal antibody responses for predicting future type 1 diabetes diagnosis, in addition to known type 1 diabetes risk factors, we can use Cox proportional hazards model that incorporate different baseline characteristics, such as age, HLA-DR haplotype, treatment arm, with respect to their performance on predicting probability of type 1 diabetes diagnosis post-randomization. And here, the metrics that we used is area under the curve, as well as partial log likelihood ratio test to compare these different models. And what we can see here is that if you look at the last line of the table, the interaction term between HLA-DR4 haplotype and the baseline IgG2 response to D. inviscis significantly improve the prediction of type 1 diabetes diagnosis compared to HLA-DR4 genotype alone. And similarly, if we use either IgG2 response to B. longum or IgG2 response to E. facalis, which I didn't talk about in this talk, they both independently improve the prediction of type 1 diabetes diagnosis prediction compared to a model with only the other known type 1 diabetes risk factors. So to summarize what I've talked about so far, we showed that in the context of a type 1 diabetes prevention trial using T-cell targeting immunotherapy, we can use anti-commensal antibody responses to identify individuals that are at risk for type 1 diabetes at an early stage, and predict those that are likely to benefit from treatment. And for future works, we're interested in looking at the relationship between anti-cancer antibody responses and T-cell immunophenotypes to gain more mechanistic insight on immune modulatory effect of tocilizumab. And finally, I would like to acknowledge all the people who have helped me in this project. My supervisor, Dr. Jane Danska, who's a great mentor and a really great scientist who provided a lot of guidance and support for this research. Our technician, Sean, who helped me complete all these long experiments with hundreds of serum samples, as well as our collaborator, Dr. Kevin Harold, who provided all the serum samples and is absolutely indispensable for this project. Thank you so much for listening, and I would be happy to answer additional questions.
Thank you, Quinn. This is a really great foray into this whole using the microbiome, which has really become more and more to the forefront in type 1 diabetes, as a predictor, or really just sort of to understand as a tool to understand the etiology of type 1 diabetes. And we have several sort of researchers in the audience here that work very closely with that. I wondered, and this is sort of a question out to the whole group. What are people's thoughts about actually, your work is showing, those, the sort of the characteristics in the microbiome of those that are best suited to respond to tocilizumab. But what about the idea of identifying, very early and the changes in the microbiome and modifying it, reseeding or resetting the microbiome? And I don't know if anyone else can speak to this. I think
I would. Yeah, I mean, I can comment on that, though we don't have like a clear conclusion yet at this point. So our lab actually is working on inoculating germ-free mice with gut microbes derived from seroconverters, those who have islet autoantibodies in their blood and non-seroconverters. And we show that these germ-free mice have difference in terms of progression to autoimmune diabetes. So I think it's actually, it's very promising that, or at least we can know for sure that gut microbiota has an effect on type 1 diabetes progression. But I think it would be a little difficult to, I guess, narrow it down to like a few strains or just a signature in the gut microbiome, because there's so many added layers in terms of human response to the gut microbiota. There might be effect at the community level, like maybe some of the gut microbes work together to bring about changes in, I guess, immune responses. There's also host difference in terms of their response to the gut microbiota. There may be different metabolites that are generated by the gut microbiota. They're different in terms of like when they are placed in an inflammatory versus less inflammatory environment. And so there are a lot of complexities that we can delve into in the field. But for now, I think we know that the gut microbiota is associated with type 1 diabetes risk to some extent. It's promising that we can modify that. It's a environmental factor that we can act on to reduce the risk of type 1 diabetes.
Yeah, it's interesting that it's there, as the paper I just brought up earlier. It's there very early. This dysfunctional signature. It appears to be, anyways, and so there's there's a lot of room for intervention and, perhaps even, teplizumab and maybe even other druggable targets that can be discovered, and then sort of, understand the microbiome's response in context. Here's a question from Kari Gerder. Is there any report on the effect of these strains in T1D? How does the IgG response affect their functionality?
Right. So there has been studies showing that these bacteria are associated with type 1 diabetes risk, but not necessarily in the same direction. Bifidobacterium longum is a very common probacteria, so you can probably buy it over the counter and take them. Although I'm not completely sure how effective it would be for modifying type 1 diabetes risk just by ingesting a single, bacteria species. In terms of the effect of IgG response affecting their functionality, these are non-pathogenic serum antibodies. So it could be that these antibodies affect their functionality, but it's more like they are produced under homeostatic states. So like they are not really causing the disease. They are more used as, I guess, a reflection of the general inflammatory tone of an individual to the gut microbiota. And IgG2 response is one of the main type of antibody that is responding to gut microbes.
Good answer. Any other questions for Quin? Well, I would say that this is a really fantastic paper, particularly for someone early in their training as a grad student. So congratulations on this really important work. And, it was great to have several, big scientists in the field here listening and communicating with you. I think there's so much to do in this field, in this part of the field. So we look forward to what'll happen next. And thanks again for joining us today, Quin.
Thank you so much for having me.
Thank you. Have a great rest of your day.